Okay, so I'm chairing this first session. So the first talk's by Erik Lindahl and on computational topics, and hopefully he's going to tell us how to restore the missing wedge.
Thank you so much, Helen. I'll get started right away. It's great to be here, but it was also a bit strange to be invited here. From one point of view, I'm a bit of a strange cat here. On the one hand, I'm nowhere near the expertise of even most of you when it comes to actual structural biology. I once swore that I would never do structural biology. I'm not doing very good on that promise. Now we're even starting to overexpress proteins. And then on the other hand, there are people like Horst and others who know the CRY-EM computational fitting, the algorithms, much better than we do. But we're somehow gradually getting in this field, and we're kind of enjoying ourselves. And I realized the beauty of having a new field is that we're all kind of beginners here. We're probably more beginners than many of you are. But we're having fun, and I'm going to share a little bit of some of the fun things we're doing, some of the things we are, in particular, that we've done together with Horst on Reliant. I'm going to try to tell you a little bit about past things in Reliant, not so much to describe algorithms and details, how we've done things on the GPUs. But I know that many of you are users of these codes, and I'm going to try to give you some tips how to actually use them better and at least understand the limitations to make sure that you make the right choices. And I'm going to speak a little bit about both current and future workflows where we would like to take things. We are super interested in feedback from you because we have some ideas where we would like to set things up in Stockholm. I'm sure you have other ideas here at both at EBIC and your local labs. I'm going to give you a little bit of hardware information and recommendations. Most of that's available online, so I'm not going to spend too much time on it. I'm going to have show you some ideas where we're working on things in Reliant 2.1, or things that might be in Reliant 2.1. And then, given time, and I'll make sure, I might have to skip some slides to make sure that I get to this Bayesian fitting where we combine cryo-EM with molecular dynamics. I think that's a very promising technique. That in turn might have some applications for how we actually look at the ensembles in cryo-EM versus simulations. So the reason I'm standing here is the following. About 2 years ago, we got money to set up a cryo-EM facility in Sweden. For us, this was the first serious facility we've ever had. We've had individual groups that have had much smaller microscopes before. But with the introduction of the new direct electron detectors and everything, we were lucky enough to get 2 major research foundations in Sweden to support us getting the microscopes. We would never have been able to do this without lots of really good friends in the UK. In particular, LNB and everybody there, they basically gave us a shopping list of everything to do and things not to do. But we made some mistakes in this process. There was just one small part that we kind of completely ignored. So actually, this is what it looked like almost exactly 18 months ago after Christmas. We got the room ready 24 hours before we got the microscopes coming in from FEI. Plenty of margin. Although it was the day after Boxing Day, so it was not entirely easy. There was only one small part we forgot, the computers. And I think Gunnar originally, when we wrote this application, I didn't really think of this I thought of this as structural biology, not computing. So when somebody, I think it was Gunnar, said, well, let's reserve 200,000 euros for it, that should be fine, right? And then when we were over at LNB in particular, started talking to people there, said that you meant 2 million, right? And that's our problem. We didn't really have the money for the computers. And that's suddenly when I realized that my job in this part is going to be to fix this one way or another. And the reason why we ended up doing this is that we've spent tons of time optimizing other codes in HPC, in particular molecular simulations codes running on GPUs. This is stuff that works that started already 15 years ago. And when you hear GPUs nowadays, today it's obvious that you can do computing on them. We started on GPUs like this one in 2001 at Stanford. Nobody had ever thought of the idea that these were actually computers. They were just meant for graphics. Now, the reason why these type of hardware is coming popular, there are lots of computer science reasons for it, but it's basically, it's going to be, if you look forward the way computers are heading, they're all going to look more like GPUs. So they're a horrible pain to program. We don't necessarily like them, but neither we nor you're going to have any choice. If you want good performance going forward, you're going to need to learn to program processors like that. And because we've gotten great performance benefits, not just we, lots of other codes too, obviously we realized that this is probably the way you could do something on cryo-EM too, and we started talking a little bit, of course. The reason why GPUs are fast is that they're completely different. When you think of a modern GPU, they look something like this. If you buy a professional card, that's a Quadro P100 card, probably cost you a bit $5,000 or $6,000. Sweet piece of hardware. The reason why they're fast is inside this chip, you have something like 3,500 small processors. Now, it can't do anything like you could do in a normal word processor or anything, because that these chips can only do one thing at a time. They require you, whatever work you have, you as a programmer have to split this in 3,500 parts. It parallelizes perfectly on all of them. Which, of course, they can't run normal programs, but if you have 1 billion pixels on a screen or something, it's going to work really well. If that was the only type of hardware, we probably wouldn't be doing this. But the wonderful part is that this entire development was driven not necessarily by these cards, but by very similar gaming cards for $500. And as important as we think science is, if you go out in the real world, what drives the market are the gamers. But that's great for us, because that pays the entire development cost and everything, and we can use those cards too, in most cases, at least with some limitations. And that's why we are seeing on the CCP-EM mailing list, there are all these discussions about these consumer cards and everything. They're outstanding value for money, more than an order of magnitude better than anything else. So the reason we started working with Course, I think there is an honest reason and a more noble one. The honest reason is that if you just look at these plots of software packages, it's kind of obvious where the flow is going, right? It's Reliant or you're dead, pretty much. And I can say that because that's, again, a year or two ago, we were not necessarily tied to Reliant. In this case, it's obvious what code we should start working on. The other thing that we've seen a lot in our case, and it's also very good code, obviously, but which is the reason why people use it. But the other case that we've seen in so many cases is that whenever you find a new code or something you would like to do, you instantly start your own code. There is lots of reasons for that. In particular, it's easy to get credit and everything, but since we're reasonably well off when it comes to funding, we figured for once we shouldn't do this, and given that we got so much help from LMV with the microscopes and everything, let's see if we can start contributing something back. And then, of course, the less noble reason is that we would have been completely handicapped unless we had all course knowledge to build on here. The reason why this works well, you have probably heard of, but I'm going to repeat it anyway, because this is important for the last piece of the talk. There are lots of optimization procedures used throughout science in general. What Reliant uses, and that's going to turn out that in general is an extremely useful way to compare models with experiments, is to use Bayes' law so that instead of having the complicated problem of determining directly optimizing a model from some sort of given image data, can be a virtually impossible problem. By using Bayes' law and normalizing with the relative probabilities here, you can kind of turn this around so that given the model A, what image data would you produce? And again, if you have a simulation or something, or at least a model, if you know all the coordinates of all the atoms, we could even calculate the micrograph. We normally don't do it, but going in that direction is trivial. Going from the micrograph to the model is a complicated problem, and this turns it around. Remember that, because I'm going to come back to it. The other neat thing is, of course, doing it this way, you can include all the experiments. You can basically simulate your entire experiment. You have this microscope CTFs, noise estimates, and everything. Any more or less crazy idea you would like to include here, that's an important piece of the experiment, you can include in your model. But to have this working right now, you need some sort of initial model, some structure to start from in this loop. And this intermediate part of the both aligning and orientation here, this gets expensive, because we have no idea what these things are initially, so we're going to need to take our model and calculate what is the probability that for this model, any way you align it or rotate it, what is the likelihood that it produced that image? And that results in lots and lots and lots of floating point operations. So that's why you have a bunch of bottlenecks, both in Reliant and other codes, that makes it expensive. So these are the ones we had to crack, one after each other. And the good part is we had a fairly good deadline. We knew that the microscopes were coming in like 12 months, and we needed a factor 10 for the same price. So thank God I had two super smart students at the time that were supposed to do completely other things. When we started talking about this, they were game instantly. Of course we're going to do that. And they had never worked with cryo-EM before, and neither had I really. But occasionally it's good to be stupid. So we just started working, and then we gradually improved, and every time we talked to course, we learned more things about things we were doing wrong or incorrect, and then you gradually improve. And at some point, this started being so good that it was like a factor 2 faster, a factor 5 faster, and suddenly we were in the ballpark of an order of magnitude. I'm going to tell you a little bit about this, but I will have to skip some of these slides in the interest of time. Reliant does most of the advanced stuff in Fourier space, and there are lots of reasons why that is more efficient, but that also means that we're going to need to treat this, we're going to need to take our models and Fourier transform them, and then we need to keep extracting and back projecting slices in Fourier space. And these operations, while they're certainly more efficient than doing it in real space, they are still very costly. So what we decided relatively early on is that we're somehow, if you start from all these input images, here we can have lots of data, but at some point we're going to need to Fourier transform over, then start calculating all these differences between the current model that I like, the Fourier transform of this model, and then extracting a small slice here. And again, this could be a more or less random rotation that I want to test. And then see this rotation of the model. How well does that overlap with each micrograph I'm seeing here? And if this is a very high probability, I will eventually start to move my reference model here so that it corresponds more to those micrographs. So this entire part we want to do on the GPU, the shaded part here. That will mean that we're going to need to send lots of data back and forth between the CPU and the GPU, because when it comes to representing all these models and doing the maximization stage, which is basically the stage where we update our model given all the probabilities, that's going to use a ton of memory, and we can't do that on the GPU. So very early on, it turned out that one of the hardest parts for us here was to find a way to just fit this data on the GPU without moving everything to the GPU. Because if we moved everything to the GPU, you would be stuck to 50 by 50 pixel images. The problem is that that is very expensive, but if there is something GPUs are awesome at, it's doing things that are floating point expensive. There is a bunch of information that you get from Reliant that tells you how expensive these things are, but I will skip those two in the interest of time. To get this to work, you always need more parallelism. GPUs can never, programs that are written for CPUs are never parallel enough. So that no matter what program you start using, your challenge is going to be expressing this parallelism. The beautiful thing with cryo-EM is that things are extremely parallel. So each image is independent of each other image, so the micrographs. You can have, for each particle here, if we now have 5, 10, or why not 50 multiple classes, you can calculate things for each class independently of the other class. For each class, you have multiple orientations. Those are also independent. And even for each orientation, you have the individual pixels in each image oriented that way as parallel. So you can have millions, if not billions, of things that are parallel here. So the GPU is going to be happy. But that doesn't necessarily mean that we are happy, because the vendors of the GPUs, of course, they try to segment the market here a bit. So they're well aware that most scientists, we care about accuracy, we want to use double precision. The problem is that double precision is only available on those expensive cards for $5,000, and we don't like that. So unfortunately, if you just switch Reliant to single precision, you're not going to get things accurate enough. So what Björn and Dari end up spending quite a lot of time on is to identify the places where we are precision sensitive and try to run only a few things in double precision and change algorithms so that they work in single precision. And to make a long story short here, this works beautifully. You can even calculate the relative deviation if you compare the CPU versus GPU versus 2 CPU runs here. The relative distribution of different image orientations that we get as a result of the refinement is basically indistinguishable. We're down to statistical noise here. For the professional cards, this makes like a factor 2 difference. For the consumer cards, you are dead unless you run single precision. The consumer cards are a factor 10 slower if you run double. You can run double, but don't do it. It's only meant for testing internally for us. Don't even think of running double. It also turns out that we can use a bunch of fancy features such as textures. If you're ever playing Doom or all these games, there are all these beautiful images, but they are actually, people cheat. So if you look here, the GPU doesn't draw the shadow in every small segment here. What the GPU does is that it draws a sphere. And, well, a sphere is actually just a circle, right? And then we fake this to be a sphere that by painting an image on it, but distorting this image to fit the shape of the sphere. This is another example where you just have a small image that you fit in a torus. So the GPUs have special hardware units to do these things. Now, that's very useful if you do games, but it's also very useful if we keep having grids that we rotate and want to project on other grids, which is everything we do in Reliant. So that the better these cards are for games, the better they are for us. And the final thing that we unfortunately had to solve is that most of these scientific applications, they don't allocate memory that much, but in Reliant we have tons of images that we need to work with. So it very quickly dawned on us that we're going to be completely limited by memory, and worse, we're going to be completely limited by the way these programs handled memory. So Björn and Dara, we had to write our entire memory allocator, the way of handling memory on the cards ourselves. Why do I tell you this? Well, because you're occasionally going to get some strange errors from this type of code. You're going to say that we could not allocate enough memory on the GPUs. Occasionally that's because the problem exists between the keyboard and chair, and occasionally it's because you're going to need a more expensive GPU. I'll get back to that in a second. So where all that coding gets you is that we're getting something, this will depend a little bit on your problem, but you're getting something that's at least an order of magnitude speed up relative to your money. The only problem is that this data is not correct. It's completely outdated. Already in the release branch of Reliant, we're something like 25% better than this now. And in particular, if you have multiple cards, we scale better. And the next version of the Reliant is going to be even better. So forget about that factor 20. The new cards, these cards are cheaper. You're not going to pay $6,000 for this. You probably pay $4,500 for this workstation. And with newer cards, the 1080 Ti cards are going to get even more performance. So this gap is going to keep growing. So it's within a year, we're probably talking about a factor 20 price performance difference. And as time goes by, this is going to be increasingly bad for the CPU. But there are some other things that I would like to draw your attention here, that some things that might appear bad, but they're actually pretty good. The first thing we see is that things don't really scale that well. This is a cheap workstation with just two very cheap cards, less than $2,000 for the entire machine. If we take that and go to a workstation that costs 3 times as much, do you see how you only get 30% improvement? The first version we released of Reliant was pretty sucky when it comes to scaling over multiple cards. And the reason for that is that when we started all this work, we spent 99% of the time in all the computational intensive parts. It was just 1% spent on maximization. Who cared about maximization? Well, the problem is that once we removed those 99%, suddenly we spent 50% of time in maximization. So the reason here is that we're suddenly bottlenecked by the 1% that was still running on the CPU. We fixed this, and I'll show you how in a second. The other thing to be aware of with GPUs is that if you're using Reliant and keep increasing your number of classes, you will use a bit more memory, but in particular, the performance is, well, the time it takes is pretty much proportional to the number of classes. So most of you typically use a handful of classes. In many cases, you don't need more than that, but a sweet thing with the GPU is that since the computations are relatively cheap, you can afford to use way more classes. We occasionally go for 10 or 15 by default, and sometimes we've tried up to 50. They don't use a whole lot more memory, and it's not going to be much slower. So don't hesitate to crank up these sliders with the number of classes. It might very well help you to see some stuff. And even with all this stuff, it's kind of embarrassing. We're still nowhere near maxing out the GPUs. If you haven't tried this yourself yet, I'm just going to show you one slide to say that when it comes to the accuracy and everything, we get, within the statistical errors, we get exactly the same result in the CPU and GPU. So this is a small segment of a ribosome on the CPU, and if I just overlay the GPU part here, it's basically identical. Again, the small statistical noise here comes from random numbers and slight differences, summation algorithms. We can't see any significant deviations between CPU and GPU parts, although the GPU side is running in single precision here. So that's kind of nice, but that was just an order of magnitude or maybe 20 times speed up. The other part that is way faster nowadays is particle picking. So I won't have time to go through all the details of particle picking either, but particle picking in Reliant uses a very simple algorithm. And I know I am well aware, and you probably have seen it too, that there are lots of publications now advocating using, say, deep learning and everything for particle picking. I would argue that you normally don't need it, but particle picking is a fairly easy problem. If you have reasonably high quality data, the way Reliant does this is that we're basically comparing We're basically taking the input templates and then comparing them in Fourier space to see to what, if we allow arbitrary rotations and alignments, what particles look like other particles. So it's simple, but it's also safe. It doesn't, if you've seen some, you might also have seen some of these things online that you can easily fool machine learning methods. They're powerful, but they can occasionally go wrong. And this is a super stable algorithm, and we have never really seen it fail. Björn came up with a smart idea that in this case, you're not really trying to refine, you're just identifying things. So we can low pass these micrographs and just use much, much, much fewer Fourier components. So this, of course, means that we're throwing away data. But even when we throw away tons of data, we're basically never, ever going to miss any components. So we typically filter to something like 20 angstrom. In this case, there was one particle we're missing. I'm not even sure whether this is a true particle, because these are supposed to be ribosomes, and that one is a bit stretched out. So we can't really see any significant difference whatsoever in the quality. Using one single GPU, and again, these are older GPUs by now, they're almost a year old. Using one single GPU is more than 100 times faster than using a CPU, and we can easily put 4 GPUs in that machine. So on a single desktop, you can pick roughly 600 times faster than you used to do before. So from our point of view, this is so fast that we've stopped optimizing this. There is no point anymore. If you need this to be more than 600 times faster, tell us. Otherwise, we're not going to spend any more time on it. So that takes us back a bit to the workflows. We already in some of the first papers we wrote to Shores, we started looking a bit that if you actually apply this in practice, how are things changing? And I'm going to show you one example from that paper, but then I'm going to show some recent stuff that some published. So in the paper, we showed that if you use beta gal, which is, at that time, was the largest data set ever collected, weeks. If you now just run this in Reliant, this looks a bit sad because you have all these large parts remaining. But the reason why these parts are large is that they used to correspond to a few % of the runtime. We used to have the case that 95% of this entire circle was yellow, and it would take you several weeks on a workstation. And we've just obliterated those computationally intensive parts. So we no longer spend the majority for the entire problem here, we spend something like maybe 20% of the time actually in Reliant. We would still like to improve that, but of course this means that suddenly improving motion refinement, polishing, and unblurring, these might very well be the part that you should start to attack next. And we've tried to see how this works in practice. So last summer, when the microscopes were brand new, and we had Alex Yammans had just come to Stockholm, we had a great new summer intern. This is Bijoya. This was her second biochemistry experiment ever. She's going to be so disappointed the rest of her career. And this is stuff in the pipeline. I'm not going to go into details about the actual protein here, but at 9 a.m., we went down and bought spinach leaves at Lidl. By noon, we had this purified, and we intentionally didn't purify it that much because we wanted to see that if we really throw a real challenge at Reliant and tell you that you're going to need to purify some horrible samples, so we included lots of fractions that we know were incorrect. During the afternoon, we started having data. We kept collecting data, and then somewhere around midnight, we started to have the first particles out. Björn and Dari had to work all night. We should automate this at some point or have more students. By 9 a.m. next morning, we had 4 angstrom structures. Now, of course, we were insanely lucky. It's not going to work this way in practice, but imagine if you could start automate, because all the computational parts here, we could, in principle, automate. And if this were automatic, by the time you push the button and start collecting, less than 24 hours later, you're going to have the results, which in so many cases will, of course, be bad, but we can't fix that. So the other thing that this leads us to think about is really how much data do you need? Do you want more data? Do you want better data? I think it's fairly common that you end up with orientations that look something like this. If this was your sample, what would you think? Is there enough diversity in this sample? How many of you think there is enough diversity? Yeah, of course is of course right. There is enough. This is going to work fine. So that we all think that you're going to need these beautiful uniform circles. There is no way you need that. Perfectly enough. what actually turns out, in many cases, we can even go the opposite way, so that we, again, having this data and that the computations were still took about as much time as the collection, we started playing around with reducing resolution so we have less noise, reducing the number of particles, and basically the more you move the slider to the left, the better it gets. So we can do fairly decent reconstructions, again, a bit over 4 angstrom with 20,000 particles. So of course, that's not necessarily what you should do for your next Nature paper, right? But don't hesitate to collect at low resolution, getting some data, and then within 1 hour, and we're talking about one single workstation, not a cluster, you can do reconstructions in an hour. And not just classifications, the complete reconstruction. I'll skip that in the interest of time. So what we've started to do in Stockholm is that we would like to automate things more and more. Right now, it's only the motion correction and GCTF that's automated. So we use SciPion for this, that most of you are well familiar with. So Jose Miguel just started in Stockholm, which is wonderful. So what we now do, again, is our way smaller facility than EBIC, is that we've centered our entire workflow around SciPion. So anytime we start a new project database and directory with SciPion. And then SciPion watches this folder, and we just tell the FEI microscopes the second you get the data, move the data over to the Linux partitions we have, and then we do motion correction, GCTF, everything automatically. You don't even have to move the mouse once. So all our users then get this finished SciPion report with their data. Of course, this doesn't necessarily do Reliant refinement, and we had some beautiful ideas that we're then going to use our cluster for Reliant refinement. Our cluster is 50% idle. Not because it's not useful, I think it's just as good or bad as most other clusters, but what I failed to realize is that everybody really loves these cheap desktops when you have 2 cards. The reason why we only have 2 cards is then they're completely quiet, and they fit under your desk. So basically, every single person in Stockholm working with Reliant has a desktop like that. They're not even $2,000 now. Again, if there is one thing you should do with all your students, get them machines like that rather than waiting for the queue, because their time is probably worth more than $2,000 over a year. So now that you know the background, I'm going to have a couple of slides with a little bit tips and tricks, but I'm going to need to speed up because we're running a little bit out of time, though we started 5 minutes late. When it comes to hardware, number one is get consumer hardware with one caveat. If you expect professional support, buy professional hardware. Basically, NVIDIA is not going to care if you come crying because you bought 500 cards and they were all overclocked that we told you not to do, and then something breaks. Yeah, that's your own fault. So I think this is a good community. You will get lots of help for this in the community, but there are some differences if you pay $5,000 instead of $500 per card. If you're happy to tinker around a bit, then you're going to be happy. Or I think there are a bunch of vendors that will even sell you machines with consumer cards, and then hopefully you can complain to them if things don't work. I would also going to say that you need to be a bit careful about new cards because there have been bugs, so don't assume that new cards work. The last few weeks, we've tested GTX 1080 Ti to the extent that we're really happy with them. So go out and buy these 1080 Ti cards if you want. They're going to be sweet. The things you should think about Reliant is that this far we've focused almost entirely on single particle. It doesn't mean that we're not interested in tomography, but I know even less about tomography than single particle, and I'm so not a single particle expert. If in doubt, spend more on the GPU than the CPU. Our GPU nodes are 50% idle. Our CPU nodes are 90% idle. We should just have gotten all GPU nodes. And then there are some things that you need to be aware of, that single precision reduces the CPU memory slightly, but not much. So you still need a lot of memory on the CPU side for the maximization. Reliant will happily use multiple GPUs and everything, but you need to think a little bit about how you set that up. I'm not going to tell you exactly how to do it because it will depend on your problem and the amount of data. But basically what happens is that as you're moving from 1 GPU to 2 GPUs to 3 GPUs to 4 GPUs, if you tweak around and try increasing the number of MPI processes, you can frequently get better performance by running many MPI processes on one card. But at some point, we're going to be limited by the parts that are not running on the GPU. And that's why the performance was a bit embarrassing in the first release. We fixed this already in the release branch. So the release branch is going to scale really beautifully over 4 or 8 cards on one nodes. And it scales well over, I think we use 4 or 8 nodes in Stockholm. It's going to scale over modest size clusters, up to 40 GPUs or so. But this leads to another problem when you start tweaking around like this, and people do this on the CCP-EM list. You start getting these horrible error messages about going out of memory on your GPUs. The reason for this is that first you only had 8 gigs on the GPUs, and when you run out of that, you run out. We can't swap from a GPU, so then we crash. The other part is you're running 2 MPI processes per GPU. You just split your 8 gigs in half, so you only have 4 gigs per card. So you need to read the documentation here and try this as best you can. There is another part that I hesitated about including here, is that if you don't understand this really well, you shouldn't do it. But internally in Reliant, to get a smooth sampling in direct space after the Fourier transforms, we pad the Fourier transforms, and it definitely improves quality. But this is also the reason why we use a lot of memory, and you can only get to a bit over 400 pixels or so. There are some options here, and I deliberately won't even go into details and explain them. Read the Reliant documentation if you absolutely want to do this. You should be aware that you are compromising quality of your data. But if this is the difference, having a gigantic sample, you just want a reconstruction to start looking at it, this can help you see something. But I so want to stress that you should never, ever use that in a publication unless you absolutely know what you're doing. It's a reason why we're not, don't take this as saying that I suggest that you should do it. But if you absolutely have to, there are ways to at least play around with it. It's simply that we haven't tested this. We know that you lose some accuracy. We just don't know how much in general. The other embarrassing side, if we're going to come into a few new things we're going to do, building Reliant, I have to confess, is not the strongest side of this. It's not entirely easy, as you also see on the CCP-EM mailing list. As of yesterday morning, we fixed this. So you might have seen that we had a beta version of this. We now have proper repositories for Ubuntu. So if you go this, there are 2 commands you're going to need to issue. There are 2 packages in this place. These are miniature packages. They're just 1 kilobyte each, and they will just tell your Ubuntu machine where the repositories are. Once you have that, just tell it to install Reliant CUDA, and everything is automatic. And in the future, every time your machine updates the software, it will also update Reliant if you have to. You can completely forget about the compiling. I intended to show you this for Red Hat too. Unfortunately, NVIDIA has broken all their CUDA 8 packages for Red Hat, so I can't do this yet. The second they fix that, which will be in a week or so, we will make sure this works for our PMs too. So that's, I think we have about 50 minutes. Good. There are some stuff that we hope to get in Reliant 2.1, but there are no promises here. As you might have seen some course tweets, Reliant, we have stochastic gradients working in Reliant. So stochastic gradient solves this problem about the initial model building. So we can do completely de novo modeling with very low resolution because at this point we just want some model. The point is not going to Nyquist or anything. You're going to need a few thousand particles downscaled, and because these are downscaled, it's going to be super quick. We're talking about minutes, maybe a quarter or so. That's both good and bad. The bad thing with that is that out of those 15 minutes, we now spend 90 to 95% on the CPU. So this technically runs on the GPU 5% of the time, and everything else we wait on the CPU. We have, if we could move all the maximization to the GPU, this would take 1 minute per iteration. So it would take 5 minutes and you would have your models, at least on 20 angstrom resolution. We have some stuff coming in Reliant 2.1 where we've improved this maximization step. So I hope that we might be able to get this down to a third of this time or so. And I think if we're talking about 5 minutes per iteration, then at least over lunch you will definitely finish it. And somewhere there I think this is going to be good enough. After that we're going to keep focusing on improving the ultra-refine. The other part that we haven't even mentioned on Twitter yet is that we have gotten some great help from Intel to start optimizing things for Intel processors. In particular, both Knight's Landing, but also classical Xeon Skylake and Broadwell processors. These will probably be cost competitive with the high-end professional NVIDIA cards. You're likely going to, well, there is no question that it's not going to be cost competitive with the $500 consumer GPUs. But the important part is that on these machines, you can have up to 1 terabyte of memory. And that suddenly means that you should be able to use these accelerations for tomography. So you're not going to get a factor of 40 times better value for money, but tomography will hopefully be way faster than it was before. And again, there's a lot of code here because we now have to rewrite all the CPU code, but with a little bit of luck, it's all going to be in Reliant 2.1. And you can also use this as a way to have vendors competing for your graces if you run a supercomputing center. The other part that we will get by improving things on this maximization is that it's kind of embarrassing that when you scale over 4 GPUs, the part running on the GPU is the dark green part here. Everything else is running on the CPU, and this question mark at the time, I think, was mostly FFTs. We didn't quite know what it was. We're basically removing the question mark entirely. That will mean that you can probably get scaling up to 100 or 200 GPUs. And at that point, we haven't started doing this yet, but at that point, you could imagine sitting in front of the microscope PC, but automatically run refinement on a cluster while you're sitting in front of the microscope PC. And at some point, you can probably imagine, give it 15, 20 minutes, and we could have complete refinement, at least seeing the classification, maybe not auto refinement. So since we have a couple of minutes left here, I'm going to spend the last 10 slides talking about my other lab here. Since we're coming from the simulation side, one thing that we started to talk about more and more, can you combine cryo-EM densities with simulations? So on the one hand, I'm not at all a simulation apologist. I think simulations are good for some things, but I think they're heavily misused. And I think they're heavily misused in the way that I think it's natural when you are a physicist by training, which I am, although I think I lost my physics card at some point. It's natural to be, you should be able to explain everything from fundamental laws of nature, right? And then you have your model and you would like to use this model to replace experiments and everything. I think it's a good feeling. It's a feeling that advances science, but in practice is frequently not the best way to study biology. In particular because it's stupid. We have all these experiments. Why on earth should you ignore the experiments just because it's fun to show that you can do it ab initio, right? So my interest here in the future is not so much basically stopping doing these long simulations you will see everywhere and rather use simulations as a tool to interpret experimental data, but do it at an earlier stage of the pipeline than we typically do it. And while I'm going to tell you a little bit about simulations here, that we can in principle use this for any model. So remember that slide I showed you about the Bayesian approach to compare a model with data. You can of course do this directly from a structure. So if you have a structure, I can calculate what is the density of electrons in different parts of space. And if I do an experiment here, how would this entire protein scatter electrons? It's not difficult. It's certainly easier to compute than the molecular dynamics simulation. And just as we know how, given that we have a density of atomic coordinates, we can then calculate how likely is it that you would give a certain 3D electron density from a simulation. And once you do that, you end up with a bunch of mathematics that I'm not going to take you through. But this is all basically based on Bayes' law, that we can then revert the process. So we use the simulation and the force field as the prior. But then, so given how, basically let the whole cryo-EM density steer the simulation in a very, very soft manner. Now, as we heard yesterday, there are MDFs, there's a bunch of programs that try to do this. I would argue that they all go wrong, and they go wrong in some very important manners. The way they typically do is that they take the electron density and invert that and use that as the target density for atoms. The problem that if you look at the dashed arrows here, this is the inverse electron. What that means is that if you're really close to your target point, you're going to get a beautiful force and slide down to your equilibrium. But in general, you're going to be far away from the target. And when you're far away from the target, you don't really have any gradient in the density, and then you're not going to feel anything at all. So what people do is that they apply very strong forces, which completely distorts the protein, and then to save your protein, you need to restrain the secondary structure, but then you can't alter the secondary structure in the refinement. So with this type of Bayesian potentials instead, we end up with super smooth, it's basically, it's called, the reason why it's called KL, the equation we end up is similar to something called Kullback-Leibler divergence, which is statistical correlation between two probability densities. But you basically end up with something that's smooth and pretty much harmonic. So we're going to get a very nice slow force gradually pushing things to the target, even from very far distances. And I think what you see down here is when we show this for some super simple two-dimensional potentials, that you get a smooth force rather than very sharp barriers. I have an example of a protein. We're going to show you this. This takes a couple of hours to run on a computer in a simulation. We haven't really bothered speeding it up. I think we can probably run it faster now. But the neat thing is I am not restraining the protein secondary structure in any way. The protein is completely free to move in a simulation with water around it, and it smoothly fits the density here. What you might have missed here, that it even crossed some local minimum here. So that helix actually moved across the first density into the second density where it should be. So you don't get stuck halfway out. And the way that works is that the Bayesian approach basically, if I have the atoms here and then the density in light gray here, rather than just going to the density, by comparing the atoms with the density here, you end up with this attractor, which is really over here. It's basically because we're calculating a quotient between them. And that is the way that the atoms are able to bypass what appears to be local minima. Well, they are local minima in the density, but they won't be local minima in the fitting potentials. This appears to have legs. When we've tested this for a bunch of proteins, this is better than any of the fitting potentials we can see. And now we're not talking about MD. We're talking about cross correlations used in Rosetta, any way of scoring how accurate a structure is compared to 3D density. This does better than all of them. I would have hoped that we would have this in GROMACS 2017 that we're going to release in about a month. It's not going to happen, but it will hopefully be out later this fall. We've even done some of this on new proteins where we have completely brand new densities. I can't tell you what protein it is, but with a fairly substantial change of secondary structure, and it really, not just secondary structure, this entire domain is moving over here. And again, we get a beautiful fit that this might have taken a morning or something to run. Again, one workstation. So if you need this to go faster, you can always run it on the cluster. So to me, this is interesting for a couple of reasons. First, I think we will be able to improve not just the 3D density reconstruction, as of course has already done so much work on Reliant, but the sad part with many of these ribosomes, we realize that the 3D density reconstruction is not now so fast, but once we have it, people have to spend months, well, at least 2-3 people during 1 month, to just sit and click and coot and try to move things to manually build the structures. As we saw yesterday, there are lots of cases where you might want to do this because you're probably smarter than a computer. But what if you could let the computer do 99% of the work and then you just look at the result and correct the computer where it was wrong? So hopefully if this works, we should be able to have the model building working fairly well within hours rather than months. And most important, not requiring any manual work. The other part that I like is that we can now start to describe, the simulation is certainly an ensemble. It's a highly approximate ensemble and everything, but of course we have an ensemble in the cryo-EM data too, right? Which we normally treat in the way of having different classes. But you could imagine that instead of, here we have the cryo data in the middle, instead of having these micrographs and classifying them in discrete classes, which we have to do now, you could imagine that taking the simulations and initially, I think we're going to need to have a large number of classes, but Reliant will hopefully allow us to have dozens, if not 100 classes, and decide that where do all of these classes appear to fall in some sort of low-dimensional space that's being visited by the simulations. Again, the beautiful thing is that we will no longer let the simulation be free to roam space, but it will only be allowed to visit the parts where the cryodensity tells us that there are actually molecules. And long-term, you could possibly even imagine classifying the micrographs as saying what parts of a molecule are close to what micrographs. So this is something that we haven't even gotten started with yet, but long-term where I think that we would like to head is that right now we tend to refine a single structure, right? But what I would like to do is that sampling a Bayesian potential in a simulation, and whether that's a simulation or some sort of Rosetta refinement or something, it doesn't really matter. You can have multiple structures that you're talking about probabilities, and then we're somehow trying to refine all of them, and ideally using all the data that we have, not just the 2-3 highest classes. It's not an obvious win, because it's one thing that I've learned is that the whole approach, just trying to collect more data is frequently a worse approach than trying to collect better data. So it might very well fail, but from the simulation side, I think it's a much more interesting approach than doing traditional simulations. I think we have 2-3 minutes left, and I want to make sure that if you have any questions, we should have some time for that too. So there are a bunch of people that we should thank here. In particular, of course, it's been a great collaboration. I hope we're going to continue this for a long time. We would be completely handicapped without Alexia and his experimental team in Stockholm. And then we've had some great help for computing, in particular from Jake and Toby. And indirectly from CCP-EM. Thanks.
Right, thanks a lot. So we have time for some questions. I should say it would be nice if you given 10% of the budget, people could advance electron microscopy as much as you've advanced the computing side. Right.
Occasionally, the necessity is the mother of invention, right? So
could you perhaps compare your approach with the maximum likelihood MD combination like Randy Reed developed? How does that compare your approach with the one used in MD maximum likelihood in X-rays?
So what approach? The approach of maximum likelihood in the fitting with?
In the refinement of structures.
Yeah.
So how does that compare with the MD maximum likelihood MD combination like in Phoenix and?
So I'm not an expert on Phoenix. I know roughly what we heard in the talk yesterday, right? Overall, I think our main approaches in this derive in the Kullback library divergence potentials, and for us they provide a very way we can fit this really smoothly, and that we can also adjust the strength of the fit. But I haven't compared Phoenix enough to say whether things are better or worse.
Well, just in terms of methods, we can talk about that later.
Yes.
Other questions?
Oh, sorry, it's a little bit too long to expand on in 30 seconds.
So some very, very cool work there. And I particularly like being able to actually overcome those local minima. It's something that is always, well, it's one of the things that got me doing the interactive work in general. But I guess it's more of a caveat than anything on that sort of large-scale fitting is to be absolutely sure that your starting model is essentially a correct but different model. In that if your starting model has large imperfections, the MD alone is never going to overcome those. An example, if you've got a tryptophan that's pointing to the wrong side of a helix it's facing, the MD will not overcome that sort of thing. So but other than that, it's really quite beautiful.
No, right, I agree. Although that's of course a general problem anytime we're starting the project, you're never guaranteed, even when it comes to stochastic gradients. If the model you get from stochastic gradients is simply too far away from Earth, you risk of only obtaining a local minimum. When it comes to MD and fitting in general, I would actually argue I'm not so worried about side chains pointing slightly in the wrong direction, because it's usually a problem you can fix just by allowing the MD simulation to run a bit longer.
Well, no,
the danger, I would say, if you start having two helices that have a rotation, or there's a packing that's assuming that you made a register offset that you packed residues the wrong way, that's going to be much harder to fix in MD, because even though you know what the gradient, which direction we should go in, once you're packed, you're not going to be able to take these residues straight through another. And even any maximum likelihood method is not going to unpack the helices and then repack them correctly. Yeah. So you're certainly right. It's very easy to get stuck.
Yes, yes. It's garbage in, garbage out.
But at least you don't always get stuck in the next local minimum.
Yeah.
Okay, any more questions? I don't see anybody. All right, we better move on. We're a bit late anyway. So thanks a lot.
Thank you so much.